Welcome to this session in which we will be covering some of the solutions for next generation sequencing in our portfolio. Here you can have a quick glimpse of what we will be covering today, starting with a brief introduction about Condalab as a company. Then we will dive into the main part of the talk, which will be focusing on RNA sequencing and specifically the SMART technology and some of its applications. This will be the denser part of the talk, but then we'll move on to lighter topics, starting with the DNA sequencing solutions and finishing off with a quick summary of some other accessories that might come in handy for your next generation sequencing experiments. So first things first, who is Condalab? For those of you who don't know us, this company was founded in 1960. We're based in Madrid and we are currently one of the main European manufacturers of dehydrated culture media for microbiology and for molecular biology. And because of our leadership in this area, we then started to distribute some international first-rate molecular biology brands in the 90s, thus expanding our portfolio. And then we became benchmarker solution providers in the biotechnology, the research areas, among others. Here you can see some of the many brands that we currently distribute, most of which focus on the fields of molecular or cell biology, and each of them brings some added value to our portfolio. And that's what enables us to produce, give you complete specialized solutions for both old and new challenges, such as cancer research, gene editing, microbiomics, and naturally next generation sequencing, for which Takara, Norgine, and Motif are some of our most valuable assets. So now moving on to the thick of it, we will be talking about some of our solutions for RNA-seq, with a special emphasis on those kits that might come in really handy if you're working with very challenging samples containing tiny amounts of RNA. So for this, I first have to introduce you to the SMART technology, which is the core of Takara's kits for RNA-seq. And this chemistry takes advantage of the ability of some reverse transcriptases to switch templates. So here's how it goes roughly. So we start off with a carefully designed primer. It could be an oligo-DT primer, a random primer, depending on the specific application. It can also contain some sequences of interest if you need to. And that's what we will be used by the reverse transcriptase to synthesize the first strand of the cDNA. And when it reaches the 5' end of the template, the RNA template, it will add some extra non-templated nucleotides, this one's circled here. So those nucleotides will be recognized by another carefully designed oligo, which will be the template switching oligo. So this will bind this sequence here, and it will be used by the reverse transcriptase to switch template, and instead of using the RNA as a template, it will use the first strand of the cDNA, and then it will synthesize the second strand of the cDNA to end up with this double-stranded molecule, also already incorporating any of these sequences that were added in the primers. So with this technology, you can capture full-length information of the template RNA in a very fast and simple way. As you see, all these sequences were introduced in a ligation-free way, and everything happens in just one tube, which minimizes the manipulation, the risk of damage and degradation and loss of your samples. It's overall a very accurate and robust and very sensitive process. You can even detect very low abundant transcripts, such as non-coding RNAs or microRNAs. And I think the most important trait about this SMART technology is its flexibility, because it can be adapted, and in fact it has been adapted for multiple purposes. You can use it for whole transcriptomics, you can use it for messenger RNA only, or even for specific RNAs of interest. And it's even being optimized for different sequencing platforms. So overall, its versatility and its reproducibility have made this SMART chemistry an industry-leading solution for next-generation sequencing. And as you can imagine, it comes in especially handy when you're dealing with low numbers of cells or cells that have very tiny amounts of RNA, because these samples are very precious, and the fact that you manipulate them only in one tube and just for very brief times It means that you don't expose it to degradation or to contamination. So here we wanted to give you a quick overview of some of the main kits in the Takara portfolio that will incorporate the smart technology. So if you're starting with single cells or equivalent amounts of RNA, you can directly resort to our single cell plus kit, which we will be covering in a minute. If you have more than one cell, though, you need to make your choice depending on the integrity and the quality of your RNA. So if you're starting with samples that have low values of quality, low green values for RNA, below 8, you will need to use these stranded kit V3 pico input mammalian. If your samples, though, have a good quality of RNA, then you can choose between these two kits, the V4 plus and the HT plus, depending on whether you're using FACS. If you do, then you can use the HT version, which is newer and also has some extra advantages that we can discuss later. So all of these kits will yield high quality Illumina compatible libraries, and we're about to see a bit more about them in a minute. So we'll start with the single cell plus kit, which is the one that will offer a higher reproducibility and sensitivity when working with single cells or with nuclei. So in this case, the smart technology will start with oligo-DTs to capture all the full-length information from the messenger RNA in the sample. In this case, you can decide to start directly with single cells. You don't need to extract the RNA before. And in the first step, with the core kit, you will produce the cDNA, and then you will produce the Illumina compatible library itself, for which the kit includes a patented module for enzymatic fragmentation and also the stem loop adapters. So all of this can be done within 8 hours, both the cDNA synthesis and the library preparation. And the kit will give you very high yields. And if you wish to improve also the sequencing power, you can even incorporate these unique dual indexes. So it all happens without compromising the sensitivity nor the reproducibility all of the kit. And you can confidently use it if you need to work with high throughput sequencing platforms such as the NovaSeq. You can even pull up to 96 samples at the same time if you wish. So overall, this is a really good solution to study point mutations, transcript isoforms, gene fusions at the single cell level. Then we'll move on to this other kit that we mentioned was ideal if you have degraded samples. So for example, we have paraffin and dead samples, which will typically have very low green values, below 8, like we said. This kit, the stranded V3 pico input kit, requires mammalian samples. You can work with 10 to 1,000 cells or the equivalent amounts of RNA. And in this case, the smart technology uses random primers. So you will be capturing the information of every RNA molecule in your sample, including the ribosomal RNA, which we will deal with later on. A very important trait about this particular adaptation of the smart technology is the fact that you can retain strand of origin information thanks to the use of these adapters, these oligos. And also in the second oligo, we incorporate a unique molecular identifier as well, which will enable you to trace the molecules and also detect any artificial duplicates that can derive from PCR amplification rounds, thus giving more robustness to the whole technology. So with the core kit, again, you would produce the cDNA, and then you will proceed with the first round of amplification, the first PCR, in which we will incorporate the Illumina adapters and any barcodes, and in a ligation-free way. And it is at this point that we will be dealing with the ribosomal RNA, which is already in the form of cDNA. So for this, the kit incorporates the ZAP-R technology. So it has some probes that will recognize mammalian cDNA derived from RNAs, from ribosomal RNAs. And then these enzymes, the ZAP-R V3 enzyme, will cleave those cDNAs deriving from ribosomal RNA, and therefore these molecules in particular won't be amplified in the next round of PCR. So finally, you will be enriching your library in those molecules of interest and leaving behind all the ribosomal-derived cDNAs. So instead of depleting the ribosomal RNA at the very beginning, when you have very limited starting material, you would do so at the very end in the form of cDNA. So it's a very elegant solution to this problem. and it can all happen within less than 8 hours. And finally, we discussed these two kits for the event of having more than one cell but good quality RNA. So we have the V4 and the HT+, which is a newer version, and it has been optimized for the use of FACS samples. So the cDNA synthesis part, the core kit, in these cases is pretty similar with some differences. For example, the HT kit is slightly more sensitive. It can accommodate as little as 2 to 100 cells. And also, the RT-PCR happens in one step only instead of the 2 steps in the V4 kit. And also, it has a dispensing solution that already incorporates some key reagents that will be needed in subsequent steps. So overall, the HT version is slightly simpler, more sensitive, and it's also automation friendly. Then the rest would be the library preparation kit, which is same for both kits. Overall, both technologies rely on oligo-DTs to capture the full-length information of the messenger RNA. This cDNA synthesis part will happen again in one single tube. And in both cases, with the library preparation kit, you will end up with very high yields of these Illumina-ready libraries, and you can also incorporate UDIs if you want to use it on RNA-seq platforms, for example. So all these kits I've mentioned so far do both the cDNA synthesis and also cover the Illumina library preparation. But if you wish, you can also acquire only the core kits that will cover just the cDNA synthesis part. And these kits are available for the SmartSeq, HT, the V4, and the single cell versions. So with the cDNA that you can generate using these kits, you can then decide to, for example, use a modified protocol, the Nextera XT, if you want to sequence on Illumina platforms. But you could also use the Ion Express library preparation kits after an enzymatic sharing step, if you want to sequence on Ion Torrent platforms. Naturally, we would always recommend the full solution that has been completed and optimized by the same manufacturer, Takara in this case, because as we mentioned, the Plus kits offer both the cDNA synthesis as well as the module with the enzymatic sharing and the library preparation kit for Illumina platforms. So that will be the most complete solution, but just so you know, you have also the versatility of ordering just the cDNA core kit. Now, moving on from the picker input kits, we wanted to give you a quick introduction to some of our solutions for immune profiling of T cells and B cell receptors, which have lately been receiving increasing attention because they're good ways to learn more about basic immunology, how the infectious diseases progress, the adaptive immune response, and also naturally the development of new therapies. But as you know, the great variety of BCRs and TCRs is so much that it's a huge challenge to analyze it. Traditionally, it's being assessed with multiplex PCR, which has certain limitations. First, the fact that it uses fixed sets of primers that are near unknown locations, which means that if there are any new variants, you won't be able to capture them with this approach. And also, as we all know, PCR is subject to great amplification bias, which can greatly distort your results when analyzing BCRs and TCRs. So to overcome these limitations, we would recommend our immune profiling kits for BCR and TCRs by Takara, which, as you can imagine, incorporate the SMART technology at the core and also coupled with the 5' race technique to capture full-length information from the TCRs or the BCRs at the RNA level. And also because it uses UMIs and also avoids the multiplex PCR, you will gain much more sensitive and robust information and a more comprehensive view of all the variants, the new functional variants that might come up. And it also comes with a free software if you want to further analyze your sequencing data. I'm not really going to go into detail about this kit, but I just wanted to give you a quick glimpse at one of the parts of the TCR kits. So this would happen after the cDNA synthesis from the TCR, and we find a semi-nested approach in this case, in which the first round of PCR will amplify the variable region as well as most of the constant region, but then the second round of PCR will cover again all the variable region, but a smaller part of the constant region, for either the alpha receptor or the beta receptor, or both, depending on your choice. It's also worth mentioning that with these TCR kits, you can decide to either sequence all the variable region, which you can do on MySeq platforms, for example, or instead you can focus on sequencing only the CDR3 region, and this we can do on any Illumina platform. Just so you know, the TCR kit by Takala, they also offer a specific version for mouse cells and another specific solution for humans, one at the single cell level and one for both populations. So just to finish up with this RNA sequencing part, we wanted to briefly present you this elegant, simple solution for small RNA sequencing. So in this case, this kit doesn't incorporate the smart technology. It hasn't been created by Takara. Instead, it's been designed by Norgine Biotech. So with this kit, you can see the overall workflow, very simple, quite straightforward. You can generate your libraries, Illumina-compatible libraries again, within 5 hours. You can start with either total RNA or with enriched small RNA, in which case you will be able to skip this size selection step with gel electrophoresis. And also, this kit incorporates a module of library preparation, library purification that uses Norgine's first-rate resting technology to capture all the DNA. And you can also pull up to 48 samples at the same time. Now we're done with the RNA sequencing part, and we will briefly talk about some of our solutions for DNA sequencing, for which I have to talk about the Reflex technology, which in this case is the core of all the DNA-seq kits created by Takara. So with this technology, you start with fragmented double-stranded DNA, which first needs to be efficiently repaired in the first step. And then we will incorporate the stem loop adapters. So these adapters are blocked on the 5' end, so they cannot really ligate each other, and that will reduce the background noise in your library. They will ligate to the double-stranded DNA, but then it will leave some nicks on the 3' ends of the molecule. So therefore, you need another step of extension, and then we will cleave the ends of the adapter so that we end up with this double-stranded normal molecules, which will then be subsequently amplified through high-fidelity PCR, where we will be incorporating also the indexes. So this is what we call the 1, 2, 3 workflow, because it happens in just one tube within 2 hours and with only 3 steps. And as you can see here, it has minimal manipulation, just 15 minutes hands-on time, again to prevent the degradation of the contamination and loss of your sample. So it will give you very high sensitivity, which with, as I mentioned, very low background, but we will still capture full complexity of your sequences. And you can apply this technology with different kits for different purposes, and it's also compatible with target enrichment afterwards. So here we just wanted to let you know which options you have for genomic DNA sequencing or cell-free DNA sequencing using this Reflex technology. So first, if you're working with genomic DNA, you need to make your choice based on the kind of fragmentation you're doing. If you're working with mechanical fragmentation, sonication, or similar, then you need to choose based on the input material that you have. If you have between 50 picograms and 50 nanograms and up to 10 microliters of DNA, you will resort to this all-time classic Reflex DNA-seq kit. But if, however, you need higher material for input, you can use this improved version, which is the HV, the high volume kit, which can accommodate between 5 nanograms and 200 nanograms and up to 30 microliters. If, however, you decide you want to do enzymatic fragmentation or you don't have a sonicator, you can resort to the latest version of the kit, which is the Reflex DNA-seq HV Plus kit. And as we said earlier, the Plus versions of Takara's kits already include that fragmentation module. Then if you're working with cell-free DNA, your first question is whether you're going to be using UMIs or not. If you do need unique molecular identifiers, then you can decide, again, based on the input, between the classic TAC-seq kit or the high volume kit, which again accommodates a higher amount of DNA and higher volume. And there's also a plus version of this kit, but it's not really necessary anymore because you're starting with cell-free DNA, which means that the DNA itself will be quite fragmented already. And then if you don't want to work with UMIs, you can quickly resort to our PlasmaSeq kit, which is another all-time classic. Okay, so all of these kits, again, will give you Illumina-compatible libraries. And just to round up these DNA sequencing part, we wanted to briefly present some of our solutions for epigenetic research. In this case, we have one kit for ChIP-seq approaches, which is this through-plex DNA-seq kit we just saw, created by Takara. And we also offer kits for ATAC-seq and for the more recent cut-and-tag-seq. In this case, both kits are developed by Active Motif, which are internationally renowned as well-known experts in epigenetic fields. So all these approaches have pros and cons. They can be even used complementarily. And I just wanted to focus on a couple of differences that you might want to bear in mind. So the first one being the input material, because as you know, ChIP-seq approaches typically require quite a lot of cells. They need to be fixed. Whereas with ATAC-seq, you can work with fresh and frozen tissues and viable cells in smaller amounts than ChIP-seq. And cut-and-tag is the one that needs the lowest number of cells to work with. Just 5,000 will do. Then the other difference between these approaches is the kind of fragmentation. Because as I mentioned earlier, if you're using the through-plex kit, you need to have done mechanical fragmentation before. But if you're going to be using any of our ATAC or cut-and-tag kits, you don't need to worry about that because there's a transposase already included in the workflow that will be the one in charge of both cutting and tagging the DNA to prepare the library. So it's a sonication-free approach in this case, which can be quite handy if you don't happen to have a sonicator, for example. And finally, there's a difference in the number of reads required to do the sequencing itself, with cut-and-tag again being the one requiring fewer reads. All of these, again, are Illumina compatible. And that will be all for the RNA-seq and the DNA-seq parts. And now we'll just briefly have a look at some other tools in our portfolio that might be quite useful when approaching a next-generation sequencing experiment. Because as you know, it's not only about preparing a library and sequencing it, but you have also had to optimize the whole workflow, because otherwise there's some crucial steps before the library preparation that can completely ruin your experiment. So the most delicate step is actually the correct collection and preservation of your samples. And I don't know, I've known some researchers who have to give up on an entire project just because, for example, the blood samples weren't put on ice straight after collecting them, or because they were sending their samples somewhere and halfway through they ran out of ice and the samples were completely degraded. And that can ruin everything. And of course, you cannot really afford that. So in order to avoid any of these problems, what we offer is a vast range of solutions developed by Norgine Biotech, which incorporate the patented preservants, which will keep your samples at room temperature and prevent degradation. And you can keep these samples between days and years, depending on the application. We have specific kits for example, stool specimens, for saliva, for blood, etc. And with any of these kits, you can forget about having to freeze your samples, or you can forget about selling them, shipping them with ice to sequencing facilities, to your collaborators, etc. This will make your life much easier, believe me. Then, of course, you need to extract the material of interest, the DNA, the RNA, and for this we also have a wide range of products by Norgine, incorporating the first-rate resin technologies. We have kits for biological fluids, we have some for environmental samples, there's one specific kit for your particular needs, really. Then you would need to make sure that you have enough quantity and also quality of your starting material. And of course, then you will proceed with the preparation of the library, for which you can use any of the technologies we've already mentioned. It's also worth bringing your attention to this Picoplex technology, which will enable you to do DNA sequencing from single cells. And we also have some options for metadynamics for Illumina. Then you will need to make sure the library is actually okay and decent. So for this, we offer some solutions, some for quantification of the libraries, one for general purposes and one specific for small RNA. And we also have a normalization kit developed by Norgine, which comes in a 96-well format. And this will help you get rid of those primer dimers that might arise during the preparation of the library. And it will also ensure that you have homogeneous amounts of DNA in every one of your libraries, so that one doesn't hijack more readings in the sequencer than the others. And of course, after this, you will proceed with the sequencing and the bioinformatic analysis, which we can also offer as a customized service through some of our collaborators. We also wanted to take this chance to introduce you to this Active Motif's Pixel multi-sample sonicator, which was recently released. It was originally created to be used in-house by Active Motif, but they saw the potential and the versatility, and they also decided to open it for the wider public, really. So this is a very robust piece of equipment that will enable you to process and sonicate through cavitation up to 96 samples at the same time and with very high homogeneity. What you see is what there is. It's very simple. It doesn't require any extra equipment. It's highly intuitive. You can control everything from this touch screen, and that's all you need, really. And also it has a very fast setup because it doesn't require any degassing steps, for example. But I think the most attractive trait of this machine is the fact that you can process up to 12 different kinds of samples with 12 different sonication protocols, one for every one of the columns in the 96-well plate. So that means you can simultaneously process either DNA, chromatin, proteins, cultures, etc., each of them with a specific condition. And last but not least, you don't require any specific consumables. You can use just 96-well plates, standard ones, and it doesn't require any particular plates or tubes, making it all overall quite economical, really. So this is just a teaser, but we will soon giving you more information about this new piece of equipment. So that will be all from me. I hope you enjoyed it. I hope you find it useful. I know it was a lot of information to take in, but just remember that we have a highly trained team of specialists at Condalab that will be more than glad to help you and assist you and help you find whatever tool in our portfolio is best fit for your needs. And we also wanted to take this chance to invite you to our next round of online meetings focusing on next generation sequencing. This is something we started doing last year, and it was a huge success. And we're hoping to make it an even better success this year in November. We will be having some technical notes and specialists talking about these kits, but also lots of clients and researchers who have already used them and will share their experience with us. So we hope to see you there.